Right.
Hello, everybody, and welcome to our module on cardiac muscle. Cardiac muscle shares many similarities with skeletal muscle. Both cardiac and skeletal muscle are striated. They both contain sarcomeres and actin and myosin and troponin and tropomyosin, and they both have T-tubules that abut the sarcoplasmic reticulum. And I talk about all these things in detail in the video on skeletal muscle. What I'm going to do in this video is focus on the differences between cardiac muscle and especially what makes cardiac muscle unique. One way that cardiac muscle differs from skeletal muscle is that cardiac muscle has dyads instead of triads. In the video on skeletal muscle, I talked about the triad shown on the screen here. A triad consists of a T-tubule down the middle, surrounded by two terminal cisternae of the sarcoplasmic reticulum. This entire structure is called a triad. In cardiac myocytes, you have a T-tubule, but it's only surrounded by a terminal cisterna on one side, not the other, and therefore this is called a dyad plasmic reticulum. This entire structure is called a triad. In cardiac myocytes, you have a T-tubule, but it's only surrounded by a terminal cisterna on one side, not the other, and therefore this is called a dyad instead of a triad. Some other differences are shown on this slide here. Cardiac muscle is involuntary. You can't make your heart beat the way you can with skeletal muscle. In addition, skeletal muscle is depolarized by motor neurons. Cardiac muscle is depolarized by pacemaker cells like the SA node. And then finally, cardiac muscle has gap junctions, which are shown in this drawing on the screen here. Gap junctions are connections between two neighboring cells so that molecules can move from one cell to the other without having to be excreted into the extracellular space. And this is very important because this is what allows depolarization to spread from one cell to another, so that once one part of the heart begins to depolarize, a wave of depolarization spreads over the entire heart. This is not true of skeletal muscle tissue. Each skeletal muscle fiber has its own motor neuron that has to tell it to depolarize. Cardiac muscle also has a different action potential. In the video on skeletal muscle, I talk about the skeletal muscle action potential, which looks like this shape on the bottom left side of the screen here. In cardiac muscle, there is a phase 2 not present in skeletal muscle. Phase 2 is caused by calcium influx into cardiac muscle cells via L-type calcium channels. This is phase 2 shown here. It's this plateau portion of the action potential that is unique to cardiac muscle, caused by calcium influx through calcium channels. Let's talk about cardiac L-type calcium channels in a little bit more detail. These calcium channels are also called dihydropyridine receptors. And they get that name because they can bind dihydropyridine calcium channel blocking drugs like amlodipine and nifedipine. And you may know that these drugs are sometimes used in the treatment of hypertension. But here's the confusing part that you have to understand. Even though the L-type calcium channels in the heart are called dihydropyridine receptors, and even though they can bind these drugs, they do so with a very low affinity. And as a result, amlodipine and nifedipine and other dihydropyridine calcium channel blockers have very little effect in the heart. Most of their effect is in smooth muscle. In contrast, non-dihydropyridine calcium channel blockers like diltiazem and verapamil have a much higher affinity for the L-type calcium channels in the heart, and therefore these are the drugs that are used when you want to block calcium channels in the heart. So you've got to imagine that you have a cardiac myocyte, and it's got an L-type calcium channel on its surface that will allow calcium to enter the cell. This influx of calcium through the L-type calcium channels is very important for contraction of cardiac myocytes. And this is a distinction from skeletal muscle. In skeletal muscle, all the calcium comes from the sarcoplasmic reticulum. Calcium influx through channels on the plasma membrane is not important. It's different in the heart. This calcium influx through these channels on the surface of the cell triggers the sarcoplasmic reticulum to release calcium via the ryanodine receptor. This is called calcium-triggered calcium release. It's unique to cardiac myocytes. And because of this phenomena, this phase 2 of the action potential is very important. Calcium comes into the cell in this phase of the action potential that triggers the sarcoplasmic reticulum to release calcium, and that's what makes the myocyte contract. Very different from skeletal muscle, where you have a motor neuron depolarizing the muscle cell, and that causes the sarcoplasmic reticulum to release calcium that leads to contraction. Let's talk about contractility. With each heartbeat, all cardiac muscle cells contract. So if the heart wants to squeeze harder and raise the contractility, it can't recruit extra fibers. That's what skeletal muscle does sometimes, but the heart can't do this. The only way the heart can squeeze harder is by sending more calcium into the cell. This will lead to more contraction. The way this is done is the heart allows more calcium entry via L-type calcium channels. That leads to more calcium-triggered calcium release from the sarcoplasmic reticulum, and this will lead to more contraction and higher contractility. In your textbooks, this is referred to as an increased calcium transient. This is just a fancy way of talking about more calcium in the cell. It's called the calcium transient because when you measure calcium concentration in a muscle cell, you see a transient rise like this. And when there's more contractility, the rise gets bigger. That's called an increase in calcium transient, and that's what leads to more contraction. So if you understand that, then it's very easy to understand what the non-dihydropyridine calcium channel blockers do to cardiac myocytes. These are drugs like diltiazem and verapamil, and as I told you before, these are the drugs that block the L-type calcium channels in cardiac myocytes. They will decrease contractility because they block calcium entry into the cell. Therefore, they are negative inotropes on the heart. They also have properties that allow them to slow conduction and lower heart rate. This is because of effects they have on cardiac pacemaker cells. But the point here is that by blocking these L-type calcium channels, you decrease contractility because there's less calcium-triggered calcium release from the sarcoplasmic reticulum. So blocking calcium channels can decrease contractility, and in contrast, activating the sympathetic nervous system can increase contractility. So let's talk about how this happens. The sympathetic nervous system works through G proteins that are coupled to sympathetic nervous system receptors. These G proteins raise the intracellular cyclic AMP levels by activating the enzyme adenyl cyclase. If you look at the bottom of the screen, adenyl cyclase converts ATP into cyclic AMP. Cyclic AMP in turn increases the activity of the enzyme protein kinase A. And protein kinase A phosphorylates calcium channels, and this leads to more calcium in the cell. And as we've been discussing, more calcium in the cardiac myocytes leads to more contraction. This is how the sympathetic nervous system raises contractility. And here's a picture just to make this more clear. So cardiac muscle cells have GS proteins on their surface. The S stands for stimulatory, and that's because they stimulate the enzyme adenyl cyclase. Adenyl cyclase raises the intracellular level of cyclic AMP that activates protein kinase A, which phosphorylates calcium channels. That allows more calcium into the cell, and that leads to more calcium-triggered calcium release, and that leads to more contraction. And this is how beta-1 receptors work. You may know that stimulation of beta-1 receptors in the heart leads to increased contractility, and this is the way it happens. The beta-1 receptors are linked to GS proteins that raises cyclic AMP levels. That leads to more calcium and more contractility. Lusitropy is the process of myocardial relaxation. This is the opposite of contractility, and it's an active process. The heart doesn't relax just by ceasing contraction. It actually undergoes an active process called lusitropy so that it can quickly relax and be ready for the next heartbeat. And increases in lusitropy always accompany increases in contractility so that when the heart beats faster, it also relaxes faster. Lusitropy is mediated by a structure called the SERCA. It's found in the membrane of the sarcoplasmic reticulum, and it transfers calcium from the cytosol back into the sarcoplasmic reticulum. This is an active process. It's an ATPase that uses ATP hydrolysis to quickly bring calcium back into the sarcoplasmic reticulum and get ready for another contraction. Because of the SERCA and the process of lusitropy, heart myocytes can rapidly contract and relax. The SERCA is regulated by a protein called phospholamban, and the sympathetic nervous system uses this regulatory protein to modify lusitropy just like it modifies contractility. Phospholamban is an inhibitor of the SERCA, and it's phosphorylated by beta-adrenergic stimulation via protein kinase A. And when it's phosphorylated, it stops inhibiting SERCA, and the SERCA can work faster to take up calcium and cause relaxation. And here's a picture just to make this more clear. Sympathetic stimulation of cardiac myocytes leads to phosphorylation of phospholamban, as shown in this picture here on the screen. This inactivates phospholamban and relieves the inhibitory effect. This allows SERCA to uptake more calcium and work faster. So the bottom line here is that the sympathetic nervous system can increase both contractility and relaxation in the heart, both by modifying the amount of calcium in the cytosol. That's the big picture take-home point. And that concludes our video on cardiac muscle. Hello, everyone, and welcome to our module on smooth muscle. Cardiac and skeletal muscle are considered striated muscle, and that's because when you look at these types of muscle tissues under the microscope, you can see striations, which means lines or markings On the screen is a picture of skeletal muscle under the microscope, and you can see little lines if you look closely in the muscle cells. Those are the striation of striated muscle. Smooth muscle, which we're going to talk about in this video, is different. It does not demonstrate striations under the microscope, in addition to a number of other differences from cardiac and skeletal muscle that we'll talk about in this video. Smooth muscle cells are components of organs. Unlike skeletal muscle, you do not find them attached to the bones of the body. You can find smooth muscle cells in the intestines, in the airways, and blood vessels, and they have a number of roles that are usually under the control of the autonomic nervous system, things that you can't voluntarily control. For example, smooth muscle helps to propel organ contents in the intestines. Very importantly, smooth muscle cells can change the resistance to blood flow in blood vessels. These cells contain actin and myosin like skeletal muscle cells and cardiac muscle cells. However, the actin and myosin function differently than in those striated muscle tissues. Some of the differences between smooth muscle cells and striated muscle are shown in this slide. First of all, smooth muscle cells do not depend on action potentials. In other words, they do not require membrane depolarization to contract. There are action potentials that can work on smooth muscle cells, but that's not the only way they contract, and that's a big difference from striated muscle. In addition, smooth muscle cells usually demonstrate slow, sustained contraction, and this is in contrast with other types of muscle cells, especially cardiac cells. As you may know, heart tissue demonstrates rapid, quick contraction to generate a heartbeat. That's very different from smooth muscle cells. And then finally, calcium does lead to contraction of smooth muscle cells, just like in striated muscle, but the way that calcium interacts with myosin and actin to generate contraction is different, and we're going to talk about that in the next few slides. In the video on skeletal muscle, I discuss actin and myosin in detail. What I'm going to focus on here is the myosin light chain, which is a very important regulatory element of smooth muscle contraction. So in the video on skeletal muscle, I talked about how actin represented the thin filaments found in muscle cells and myosin represented the thick filaments. Well, it turns out that myosin is made up of both heavy and light chains, and the light chains are modified in smooth muscle to control contraction. So because myosin are the thick filaments in muscle cells and myosin light chain is what's modified in smooth muscle cells to control contraction, smooth muscle is said to be thick filament regulated. And this is in contrast to skeletal muscle, which is thin filament regulated. In skeletal muscle, tropomyosin is modified on thin filament so that muscle contraction occurs. It's different in skeletal muscle. Skeletal muscle is thick filament regulated. The phosphorylation of myosin light chains is a major regulator of smooth muscle cell contraction and smooth muscle tone. The reason for this is because only phosphorylated myosin light chain will interact with actin. This is a special feature of smooth muscle cells. So by controlling the amount of phosphorylation of myosin light chains, a cell can control the amount of contraction or tone. There are two enzymes that participate in this process. Myosin light chain kinase phosphorylates myosin, and myosin light chain phosphatase dephosphorylates myosin. And here's a picture just to make this more clear. So myosin light chain, when it's phosphorylated, leaves controlling the amount of phosphorylation of myosin light chains. A cell can control the amount of contraction or tone. There are two enzymes that participate in this process. Myosin light chain kinase phosphorylates myosin, and myosin light chain phosphatase dephosphorylates myosin. And here's a picture just to make this more clear. So myosin light chain, when it's phosphorylated, leads to contraction. When it is dephosphorylated, that leads to relaxation. Myosin light chain kinase will phosphorylate myosin light chain, and myosin light chain phosphatase will dephosphorylate myosin light chain. Calmodulin is a smooth muscle cell protein. It's ubiquitous, meaning there's lots of it inside of smooth muscle cells. Its role is to bind calcium, and once calcium is bound to calmodulin, that activates myosin light chain kinase, which, as we just saw on the last slide, leads to contraction. So what this means is that the big picture point is that anytime you get more calcium inside smooth muscle cells, that will lead to more contraction via this mechanism shown on the slide. I've added calcium and calmodulin to our picture here to make this more clear. When calcium and calmodulin interact with myosin light chain kinase, they make the enzyme more active. That leads to more phosphorylation of myosin light chain, and that leads to contraction. When there isn't much calcium around, then myosin light chain kinase is less active and there's less contraction. And remember, calmodulin is ubiquitous, so it's always present. So what this means is that the big picture is the more calcium you put into a smooth muscle cell, the more contraction you will get. So now that we understand that calcium leads to contraction of smooth muscle cells, let's talk about where the calcium inside of smooth muscle cells comes from. Well, first of all, smooth muscle cells have a sarcoplasmic reticulum like striated muscle cells, and the sarcoplasmic reticulum stores calcium, and that's one source of calcium for vascular smooth muscle cells. But the other important source are L-type calcium channels. These are calcium channels found on the surface of a smooth muscle cell. They allow calcium to enter the cell. Now, these calcium channels also bind structures known as dihydropyridine. So for this reason, these L-type calcium channels are sometimes also called dihydropyridine receptors. If you look at the bottom left side of the screen, this chemical structure is a pyridine. If you add two hydrogens and remove one of the double bonds, you get a dihydropyridine. And structures that contain a dihydropyridine ring bind to L-type calcium channels. And for that reason, these calcium channels on smooth muscle cells are also called dihydropyridine receptors. In the cardiology section in the video on hypertension drugs, I talk about calcium channel blockers that are dihydropyridine drugs. These drugs all end in ipine, like amlodipine, felodipine, nicardipine. All of these drugs are L-type calcium channel blockers, and they're called dihydropyridine drugs because the drugs have a dihydropyridine structure within them. For example, the structure of amlodipine is shown at the bottom right side of the screen here. It contains a dihydropyridine ring. Therefore, it is called a dihydropyridine calcium channel blocker. And if you administer these drugs to patients, it will block the L-type calcium channels in smooth muscle cells. This will lead to less calcium into the cell and therefore relaxation. And these drugs are used to cause relaxation of smooth muscle cells in the vasculature and therefore lower blood pressure in patients with hypertension. Now let's talk about smooth muscle tone regulation. There are two major regulators of the amount of contraction or tone in smooth muscle, and we've already talked about both these things. That's the amount of calcium in the cell and the degree of phosphorylation of myosin light chain. And if you remember these two facts, it is very high yield. You can answer lots of board questions just by knowing these two elements that change smooth muscle tone. Now, both of these things, the amount of calcium in the cell and the degree of phosphorylation, can be modified by various factors to alter smooth muscle tone. For example, the autonomic nervous system often modifies these factors to change smooth muscle tone, and there are local factors like local vasodilators that may modify these two things to change the tone in smooth muscle. The way that local factors and the nervous system modify the calcium in the cell or the degree of phosphorylation is by working through second messenger systems and G protein. So let's talk about these things now. There are three major second messengers used in smooth muscle cells. Those second messengers are cyclic AMP, cyclic GMP, and inositol triphosphate, or IP3. Their chemical structures are all shown on the screen here. Cyclic AMP and cyclic GMP are just modified versions of nucleotides, and IP3 is the structure on the bottom right side of the screen. These three second messengers are going to modify the levels of those controllers of smooth muscle tone we talked about before. That's how they work. So IP3 works on calcium. Inositol triphosphate triggers calcium release from the sarcoplasmic reticulum, and this leads to contraction. Cyclic AMP and cyclic GMP work on myosin light chain phosphorylation. Cyclic AMP inhibits myosin light chain kinase, controllers of smooth muscle tone we talked about before. That's how they work. So IP3 works on calcium. Inositol triphosphate triggers calcium release from the sarcoplasmic reticulum, and this leads to contraction. Cyclic AMP and cyclic GMP work on myosin light chain phosphorylation. Cyclic AMP inhibits myosin light chain kinase. This leads to relaxation. I'll show you a picture of this in a minute. And cyclic GMP activates myosin light chain phosphatase. This also leads to relaxation. But the big picture point here is that all these second messengers and all these systems work through the same two controllers of smooth muscle tone. That's calcium and the degree of phosphorylation of myosin light chain. If we go back to our picture, I've now added in these second messengers. So cyclic AMP inhibits myosin light chain kinase. That leads to less phosphorylation of myosin light chain and therefore relaxation. Cyclic GMP activates myosin light chain phosphatase, but that has the same effect. It leads to less phosphorylation of myosin light chain and therefore relaxation. IP3 is the only second messenger that leads to contraction. It triggers calcium release from the sarcoplasmic reticulum. And as we talked about earlier, anytime there's more calcium around, there'll be more calcium and calmodulin to interact with myosin light chain kinase, and that will lead to more phosphorylation of myosin light chain. Once you understand how these second messenger systems affect smooth muscle, it's then very easy to understand how various hormones and neurotransmitters modify smooth muscle tone in the body. For example, norepinephrine and epinephrine exert their effects by modifying intracellular levels of IP3 and cyclic AMP in vascular smooth muscle, and that's how they modify smooth muscle tone. Vasopressin works through IP3, adenosine works through cyclic AMP, and prostaglandins work through cyclic AMP as well. So you can read lots of basic science literature on all these systems, but the big picture point that you want to remember is the way these neurotransmitters and hormones work is by modifying second messenger systems inside of vascular smooth muscle, and that's how they modify smooth muscle tone in the body. So now we've talked about how calcium and myosin light chain phosphorylation can modify smooth muscle tone. We've also talked about the 3 second messengers that can affect calcium levels and degree of phosphorylation in smooth muscle cells. The last thing we need to discuss is how a local signal or a neurotransmitter activates these second messenger systems. In order to understand that, we've got to talk about nitric oxide and G proteins, because that is the link between a local signal and a neurotransmitter and the second messenger systems found in smooth muscle cells. Nitric oxide is also called endothelial-derived relaxin factor, or EDRF. And if you remember this name, it's a very easy way to remember what nitric oxide does. It's synthesized by endothelial cells from the amino acid L-arginine. This is the structure of arginine on the screen here. Nitric oxide is just a nitrogen bound to an oxygen, and you can see that arginine has lots of nitrogens and oxygens in it that can be used to make nitric oxide. At the basal level, endothelial cells always produce some amount of nitric oxide, but there are many stimuli for increased production. These include the amount of blood flow or shear stress on the endothelial cells, also other substances like acetylcholine, bradykinin, and substance P. All of these things can cause endothelial cells to make more nitric oxide. Once nitric oxide is synthesized by endothelial cells, it diffuses into neighboring smooth muscle cells. And once inside those cells, it activates the enzyme guanylyl cyclase. What guanylyl cyclase does is convert GTP to cyclic GMP, as shown at the bottom of the screen here. And we've already talked about what cyclic GMP can do. It's one of the major second messengers. It activates myosin light chain phosphatase, as we just showed a few slides ago, and that leads to relaxation. So therefore, the net effect of nitric oxide, once made by endothelial cells, is to diffuse into smooth muscle cells and cause relaxation. Let me make a very high yield point about nitric oxide. These substances, and in particular acetylcholine, work indirectly on smooth muscle cells through nitric oxide. They require endothelial cells for their role in relaxation of smooth muscle. That's because they trigger the endothelial cell to make nitric oxide. So if you take the endothelial cell away, then acetylcholine will no longer be able to relax vascular smooth muscle. And that's the kind of thing that comes up in board questions, this indirect mechanism of acetylcholine endothelic oxide. Let me make a very high yield point about nitric oxide. These substances, and in particular acetylcholine, work indirectly on smooth muscle cells through nitric oxide. They require endothelial cells for their role in relaxation of smooth muscle. That's because they trigger the endothelial cell to make nitric oxide. So if you take the endothelial cell away, then acetylcholine will no longer be able to relax vascular smooth muscle. And that's the kind of thing that comes up in board questions, this indirect mechanism of acetylcholine and other substances that work through nitric oxide. There are also a couple of drugs that work via nitric oxide. Nitroglycerin is a vasodilator. It's used in angina and heart failure. It's got the structure shown at the bottom of the screen here, and you can see that it contains lots of nitrogens and oxygens, and it's converted to nitric oxide in smooth muscle cells, and once this happens, the smooth muscle cells relax, and that's how nitric oxide works as a vasodilator. Nitroprusside is a drug used as a vasodilator in hypertensive emergency. It contains nitric oxide, so it's a direct nitric oxide donor, and it administers nitric oxide to the body and the smooth muscle cells and leads to very rapid vasodilation. That's why it's used for hypertensive emergency. The other way second messenger systems are activated is through G proteins. G proteins are found on the surface of smooth muscle cells. They can be activated by neurotransmitters like epinephrine, and they transmit signals to smooth muscle cells by working through smooth muscle cell second messenger systems. There are a number of subtypes of G proteins. There are GS proteins, GI proteins, and GQ proteins. The GS proteins lead to smooth muscle relaxation. They do this by raising the level of cyclic AMP. And as we talked about earlier, cyclic AMP is a second messenger that inhibits myosin light chain kinase and leads to relaxation. The GI proteins do the opposite. They decrease the level of cyclic AMP in smooth muscle cells. This leads to contraction. And then finally, the GQ proteins increase the amount of IP3. And as we talked about earlier, IP3 stimulates calcium release, and that leads to contraction. Here's a drawing of the GS and GI systems and how they work in vascular smooth muscle. The GS proteins get their name because they stimulate or activate adenyl cyclase. This leads to conversion of ATP into cyclic AMP, and cyclic AMP inhibits myosin light chain kinase, just like we talked about before. That leads to relaxation. I've written this at the bottom of the screen. The GS proteins stimulate adenyl cyclase. That leads to relaxation. The GI proteins get their name because they inhibit adenyl cyclase. Therefore, they have the opposite effects. They reduce the amount of cyclic AMP in the cell. So inhibition via the GI proteins of adenyl cyclase leads to contraction. It's very hard to remember all this. Just remember the big picture here, that the G proteins are part of how signals are transmitted to smooth muscle via second messenger systems. And here's a drawing of how the GQ systems work. GQ proteins activate an enzyme called phospholipase C, which synthesizes IP3, which, as we just talked about, is one of the major second messengers. It activates the sarcoplasmic reticulum to release calcium, and that leads to more contraction. So GQ proteins lead to contraction of vascular smooth muscle. I'll finish this video with this slide to remind you that G protein systems serve as a link between the autonomic nervous system and vascular smooth muscle. Shown on this table are a number of receptors used by the autonomic nervous system, and you may be familiar with these receptors like alpha receptors and beta receptors and muscarinic receptors. Also shown on the table is the G protein class that is usually linked with each receptor. So for example, you may know that stimulation of alpha-1 receptors usually leads to vasoconstriction, Well, the way alpha 1 receptors achieve this effect is by activating GQ proteins on vascular smooth muscle. And as we just talked about, activation of GQ proteins leads to constriction of vascular smooth muscle. That will increase peripheral resistance and raise the blood pressure. And that's why alpha 1 receptors are vasoconstrictors. You can also see on this slide the G protein class linked with other types of autonomic nervous system receptors. And that concludes our video on smooth muscle.
Bro thinks he's in a podcast. Bro thinks he's in a podcast, bro.
He's building 5 assets. मैं घंटे बोलेगा तो पेट्रोल मिलेगा, तो मैं घंटे करूँ तो म्युनिसिपैलिटी को पानी मिलेगा. Vision between Dubai real estate and Mumbai real estate. Stamp duty 7%. Only 4%. Return on investment 2 to 4%. Minimum ROI is 8%. A lot of people don't know this. A billionaire would never invest 1 million dirhams on a single apartment. Let's say he had to put a down payment of 20%, he would get 5 1 million apartments and pay 20 for each, and the rest mortgage would be paid by the tenants. Now what happens with this is, within how many ever years, he's building 5 assets. This is what the top real estate investors do. A lot of people don't know this. A billionaire would never You say you love me. Bro, it's these type of motherfuckers that I've noticed, like they'll not do well in school or college, and then when they graduate, they'll make a podcast and be a real estate agent and make videos like this. Like, and they'll try and scam you as much as possible.
serious, bro.
Hello, everyone, and welcome to our module on bone. Let's start by talking about different types of bones. The long bones are bones in your body that support weight and allow movement. These are the bones that are found in your legs and in your arms. Flat bones are bones that protect organs. For example, your skull contains a number of flat bones. Short bones are the bones of the wrists and the ankle, and there are irregular bones, which are bones found in the vertebrae. You may know that the vertebrae have a very unusual and irregular shape. And then finally, sesamoid bones are bones that are embedded in tendons, and the classic example of a sesamoid bone is the patella or the kneecap. Now let's talk about the macroscopic structure of bones. The periosteum is the outer membrane that covers the outer surface of bones. It contains blood vessels, it contains sensory nerves. If you look at this picture on the screen here, the periosteum is this outer layer here seen surrounding the long bone, and it contains the blood vessels. Cortical bone is the outer surface of the bone. It's also called compact bone. Anytime you hear the word cortex or cortical, you should just think of shell. Basically, the outer shell of anything is the cortex. For example, the cortex of the kidney is the outside of the kidney, and in the same manner, the cortex of the bone is this hard outer layer of bone that surrounds the entire bone. It's the hard exterior bone. In addition to the cortical bone, there is another type of bone called trabecular bone, or it's also called cancellous or spongy bone. This is the bone tissue shown in this part of the bone in this drawing on the screen here. This bone is soft and flexible, and it's found at the ends of long bones. It's trabeculated, which means it's sort of honeycombed with lots of spaces made by the bones. And what's important to understand about trabecular bone is that it has lots of surface area. Because the bone is curved, like drawn in this picture here, there's lots of surface area. This will be important later when we talk about cells found on the surface of bone. And then finally, the medullary cavity, or the medulla of the bone, is the part of the bone that contains the marrow. It's usually found in the shaft of long bones. And you can see some of these basic bone structures on an x-ray, and they like to show you x-rays in the step 1 exam, so you ought to be able to find the basic structures of a long bone. For example, shown on the screen is a picture of the femur bone in the leg, which is a classic long bone. Note that it has this black section down the middle of the straight portion. That is the medullary cavity that contains the bone marrow. On either side of the medullary cavity, there are these thick white portions. That's the cortical bone. And then if you look up towards the top near the joint, you see that that black part down the middle has gone away, and instead you have these straight wispy lines. That is the trabeculated bone, and as I said before, it's usually found at the end of long bones, such as up towards the joint. There are some important terms related to long bones you should be familiar with. The epiphysis of a long bone is the end of the long bone. It's usually covered by cartilage. If you look at this drawing on the screen here, the epiphysis is here at the bottom and up here at the top. The metaphysis is the widening just before the epiphysis, so that's this portion right in here. And then finally, the diaphysis is used to describe the straight portion of a long bone, or the shaft that contains the medullary cavity. Let's talk about the types of cells you can find in your bones. Osteoblasts are the cells that
Man, you can't call back or what? Say, Anamo, are you free tonight? You want to go for dinner? You, me, Rohit. சரி எங்கேயாச்சும் வெளியே போகலாம் சாப்பிட. கம் அரவுண்ட். சரி நான் என்ன டைம் சொன்னாலும் நீ ½ hour லேட்டாக தான் வர போகிற. இப்போ நான் 7:30 வா? ஓகே பை. என்னது வீக்கெண்டா? ஏன் இன்னைக்கு என்ன பிளானு? வாலிபால் முடிச்சுட்டு ஜிம்மா? ஜிம் எப்போ முடியும்? 9:30 10 ஆகிடும். ஜிம்லாம் இல்லைடா, சாப்பிட்டுட்டு சரி ஓகே நான் வீக்கெண்டே போகிறோம். ஓகே பை.
that synthesize bone matrix. We'll talk more about these cells in a minute. Osteoclasts are specialized macrophages. They're derived from circulating monocytes, and they secrete acid and protease enzymes that dissolve bone matrix. So it's an important principle to understand that your bone is constantly undergoing remodeling. There are osteoclasts that break it down, and there are osteoblasts that remake it all the time. It is not a static structure. The main two cell types to know about are the osteoblast and osteoclast, but there is a third type of cell found in bones. It's called an osteocyte. These are osteoblasts that have become buried in the bone matrix. They become osteocytes, and they play a role in controlling local calcium and phosphate levels. But the main two cell types that control bone turnover and bone remodeling are the osteoblasts and osteoclasts. So one of the things you can find in bone tissue are the cells that we talked about in the last slide, but lots of your bone is made up not of cells, but of something called bone matrix. This is the extracellular component of bone. It's synthesized by osteoblasts, the bone forming cells we talked about in the last slide. And lots of your bone matrix consists of type 1 collagen. In the cell biology section in the video on connective tissue, I talk in detail about collagen, and it's type 1 collagen that you find in bone. The other major component of your bone matrix is hydroxyapatite. This is a crystalline structure that contains calcium and phosphorus. And 99% of your body's calcium and 85% of the body's phosphorus is found in bone, and it's found in hydroxyapatite, which is made by the osteoblasts. When osteoblasts want to lay down new bone matrix, they first make something called osteoid. This is non-mineralized bone matrix. It's mostly proteins. It's laid down by osteoblasts, and later it becomes mineralized, which means calcium and phosphate are added to the osteoid. This will be very important when we talk about bone disorders, because there are bone tumors and other bone disorders where osteoid is made in excess, and you need to understand that this is a proteinaceous material that is made by osteoblasts but has not been mineralized yet. Now we're going to talk about something that is incredibly high yield, and that is bone turnover. As I mentioned before, bone tissue is not static. It's constantly changing. Osteoclasts break down old bone, and osteoblasts lay down new bone. And there is a balance between formation and breakdown. And if this balance is normal, you will have normal bone structure and strength. But if there's too much breakdown, the bones will become thin. And if there's too much bone formation, the bones can become abnormally thick. What you need to understand is that this entire system of bone turnover is modulated by signals from the osteoblast. The osteoblast is really the controller of bone turnover, and the osteoclast is really just a slave to the osteoblast. These signals, which come from the osteoblast, some of them stimulate osteoclasts, some limit osteoclasts, and the combination determines the rate of bone formation and breakdown. The first signal from the osteoblast to the osteoclast that we're going to talk about is RANK and RANK-L. So RANK stands for receptor activating nuclear factor kappa beta. This is a big, long, confusing name, but basically RANK is just a receptor, and it's expressed on the surface of osteoclasts, as I've shown in my picture at the bottom of the screen here. When this receptor binds to something called a ligand, this leads to synthesis of nuclear factor kappa beta in the osteoclast, and that stimulates the osteoclast. So what binds to this receptor called RANK? Well, the RANK ligand, or RANK-L. This is receptor activating nuclear factor kappa beta ligand. A ligand is something that binds to a receptor, and this one binds to RANK, and it's expressed on the osteoblasts as shown in my picture here. So when osteoblasts make more RANK-L, they can bind that RANK-L to the RANK on osteoclasts, and that is one way that osteoblasts can signal osteoclasts and create more bone breakdown activity by the osteoclasts. Two other ways by which osteoblasts control osteoclasts are shown in this slide. The first one is osteoprotegerin, or OPG. This is a decoy receptor for the RANK ligand. It's made by osteoblasts, and it binds the RANK ligand and prevents the RANK ligand from binding to RANK on osteoclasts. Osteoblasts can also make MCSF, which is macrophage colony stimulating factor. Osteoblasts can secrete this, and this will stimulate osteoclasts. So I've summarized everything with this picture on this slide. And basically, if you know this picture, you can answer lots and lots of questions about bone pathology and bone physiology. So osteoblasts make RANK-L, and they also make MCSF, and both of these things can stimulate osteoclasts. In addition, osteoblasts make OPG, which is a decoy receptor. It will bind to RANK-L and block it from binding to RANK. So when osteoblasts make more OPG, that decreases osteoclast activity. And I've summarized all this at the top of the slide. More RANK-L or MCSF from osteoblasts leads to more osteoclast activity. More OPG from osteoblasts leads to less osteoclast activity. Now let's talk about how bone forms in utero. When a baby is born, that baby has long bones and flat bones and other bones. So how did those bones get there? There are two major ways by which this occurs in utero. The first one is called endochondral ossification, and the second one is membranous ossification. Endochondral ossification occurs during embryogenesis. This is the process by which our long bones form. They develop from hyaline cartilage that is secreted in utero from chondroblasts and chondrocytes. That's why it's called endochondral ossification. The way the bones are made is that a cartilage mold is first formed. These cartilage molds of our bones are called enlogen. For example, a long bone might start looking like a cartilage mold like this one on the screen here. As the mold grows, the chondrocytes die, and then osteoblasts come in from the blood, and they will turn this cartilage mold into a bone. The conversion of the cartilage mold into bone starts at two places. For example, if you have a bone that looks something like this, a typical long bone, at the center of the bone, you will have what's known as the primary center of ossification. This is where osteoblasts will begin laying down bone matrix. At the ends of the bone, which is called the epiphysis, you will also get a center of ossification. This is called the secondary center of ossification. These two centers of bone formation will grow towards one another, and they will eventually meet and trap a layer of cartilage in a line like this. That layer of cartilage will become the epiphyseal or growth plate. You may know in the bones of children, there's bone tissue with two plates at the ends, which are cartilage plates, and those are the remnants of the hyaline cartilage mold that was present in utero. And here's a picture from the web just to make this very clear in your mind. So we begin with a hyaline cartilage model, then the primary center of ossification opens up in the middle of the bone. Secondary ossification centers open up at the two ends of the bone, and those two ossification centers grow towards one another until we're left with a layer of cartilage that is trapped. That's called the epiphyseal plate or the growth plate. So now let's talk about the growth plates, which are also called the epiphyseal plates. These are found at the ends of long bones between the metaphysis and the epiphysis, which are those portions of long bones we talked about earlier. In fact, the word physis means growth plate. If you look at this X-ray image from the arm of a child in the bottom right side of the screen here, you can see the growth plate right here. These growth plates contain hyaline cartilage, and they have chondrocytes which grow towards the epiphysis. So the chondrocytes are going in this direction away from the center of the bone. The osteoblasts are laying down bone matrix towards the center of the bone, and as the child grows, the growth plate will move towards the joint, with bone being laid down behind the growth plate towards the center. The bone. Now at puberty, the growth plate closes and the epiphyseal lines will be formed, which will be present throughout adulthood. So if you look at this picture at the top of the screen, which is from the knee, you can see this line right here. That's the epiphyseal line. That's the remnant of what was the epiphyseal plate before puberty. We'll get back to the in utero formation of bone tissue in a minute, but let me pause and mention woven and lamellar bone. Woven bone is the first type of bone formed by osteoblasts. It's also called primary or immature bone, and it contains disorganized collagen fibers, and it's relatively weaker bone. Later, this woven bone is remodeled or replaced by osteoblasts, and it becomes lamellar bone. Lamellar bone is layered and organized and stronger. So at the growth plates in children, you will initially see osteoblasts laid on woven bone, and then later it will become lamellar bone. You can also see woven bone in adults. If there's an injury, this is the type of bone that osteoblasts will initially lay down at the site of a bone fracture or injury. So back to our discussion of the in utero formation of bones, the second process by which bones are formed in utero is called membranous ossification. In this type of bone formation, bone matrix is formed directly and not from a cartilage mold. Osteoblasts will lay down woven bone, they'll later remodel it to lamellar bone. And this is the way most of the flat bones in our body are formed. For example, the calvaria, which is the skull bones, many of these are formed by membranous ossification, same thing for many of the facial bones. If you understand endochondral ossification and of the woven bone in adults, if there's an injury, this is the type of bone that osteoblasts will initially lay down at the site of a bone fracture or injury. So back to our discussion of the in utero formation of bones, the second process by which bones are formed in utero is called membranous ossification. In this type of bone formation, bone matrix is formed directly and not from a cartilage mold. Osteoblasts will lay down woven bone, they'll later remodel it to lamellar bone. And this is the way most of the flat bones in our body are formed. For example, the calvaria, which is the skull bones, many of these are formed by membranous ossification, same thing for many of the facial bones. If you understand endochondral ossification and growth plates, then it's very easy to understand achondroplasia. This is the most common cause of dwarfism. It is a genetic disorder caused by a gene mutation in the fibroblast growth factor receptor 3 gene. 80% of cases are due to a spontaneous mutation, but this can run in families. And when it does, it is autosomal dominant. By that, I mean that anyone with one copy of the gene will develop dwarfism. If you have two copies of the gene and you are homozygous, you will not survive. Babies who are homozygous are stillborn. So if you see a patient who has dwarfism from achondroplasia, that patient must be heterozygous and have a single abnormal copy of the gene. If they were homozygous, they would not be alive and seeing you in clinic. Basically, achondroplasia is a disorder of defective endochondral ossification. The mutation that occurs in the gene is a gain of function mutation, so the growth factor receptor is activated and turned on. Now, you might think a turned on growth factor would lead to more growth, but in fact, what happens is chondrocyte proliferation is inhibited. As a result, the long bones do not grow. So people with achondroplasia are dwarfs, which means they look like this man in the picture. He has very short arms and very short legs, but his torso and his face are normal. And that's because most of the bones in the torso and face do not develop by endochondral ossification, but the long bones of the arms and the legs do. This is a very simple disorder to understand if you just understand endochondral ossification. In the biochemistry section in the video on the lysosomal storage diseases, I talk about a group of disorders called mucopolysaccharidoses. These are disorders like Hunter's syndrome and Hurler syndrome, where children have an enzyme deficiency and they cannot metabolize mucopolysaccharides like heparan and dermatan sulfate. The reason I'm mentioning this here is because chondrocytes are cells that degrade lots of mucopolysaccharides. So in children born with one of these disorders, the mucopolysaccharides accumulate in the chondrocytes and the chondrocytes die. So as you can imagine, this leads to similar problems to achondroplasia, problems at the growth plate and with growth of long bones. And short stature and malformed bones are common in these disorders. In the basic science laboratory, when researchers are studying osteoblasts, there are a number of commonly used activity markers that are studied. These are substances whose levels rise when osteoblasts are more active. These are also sometimes used clinically to make the diagnosis of a disorder in which osteoblasts are overactive. The first activity marker is alkaline phosphatase, which I'll talk about in the next slide. The second one is osteocalcin. This is the major non-collagen protein found in bone matrix. Most of the bone matrix is collagen, but the major non-collagen protein is osteocalcin. So when its levels go up, that means basically to make the diagnosis of a disorder in which osteoblasts are overactive. The first activity marker is alkaline phosphatase, which we'll talk about in the next slide. The second one is osteocalcin. This is the major non-collagen protein found in bone matrix. Most of the bone matrix is collagen, but the major non-collagen protein is osteocalcin. So when its levels go up, that means osteoblasts are working. Collagen used long bones, and short stature and malformed bones are common in these disorders. In the basic science laboratory, when researchers are studying osteoblasts, there are a number of commonly used activity markers that are studied. These are substances whose levels rise when osteoblasts are more active. These are also sometimes used clinically to make the diagnosis of a disorder in which osteoblasts are overactive. The first activity marker is alkaline phosphatase, which I'll talk about in the next slide. The second one is osteocalcin. This is the major non-collagen protein found in bone matrix. Most of the bone matrix is collagen, but the major non-collagen protein is osteocalcin. So when its levels go up, that means osteoblasts are working harder. The third activity marker is type 1 procollagen. So in the cell biology section in the video on connective tissue, I talk about the synthesis of collagen, and I talk about how it's initially synthesized as 3 pro-alpha chains combined together. This is called procollagen. It looks like what I've shown on the screen here. This is what is secreted from osteoblasts, and then it is modified extracellularly into tropocollagen and collagen. So you can imagine that when the levels of type 1 procollagen go up, that means more pro-alpha chains are being secreted from osteoblasts, and therefore that is a good activity marker of osteoblasts. Alkaline phosphatase is an enzyme that's found in bone tissue and in the liver, although there are different forms, and levels of alkaline phosphatase in the blood can rise in bone disorders or liver disorders. There's also a placental form called appropriately placental alkaline phosphatase, and you can see high levels of this in the blood in some germ cell tumors. The reason this is important in bone is because it's one of the major proteins found in bone tissue. It's found bound to osteoblasts and also free. And what it does is it creates an alkaline environment around the osteoblast. And if you can remember that alkaline environments are favorable for calcium deposition, that's very helpful to understanding bone physiology. The osteoblast creates a high pH alkaline environment around the cell, and that makes a favorable environment for calcium to settle out and form crystals of hydroxyapatite in bone. And if you can remember that an alkaline environment is favorable for bone formation, that's an easy way to remember that acidosis is bad for bone formation. An acidotic environment is unfavorable for the deposition of calcium crystals. In addition, acidosis stimulates osteoclasts, and you can see hypercalcemia as a complication of chronic acidosis from bone breakdown. And patients who have a chronic acidosis can also develop reduced bone mineral density and osteoporosis. And this sometimes complicates patients who have renal tubular acidoses. These patients are chronically acidotic, and that can have negative effects on bone formation. I talk about parathyroid hormone in detail in the endocrine section in the video on the parathyroid gland, but I'll review its role in bone physiology here. So parathyroid hormone has multiple effects on bone. It can stimulate bone resorption and formation. The dominant effect varies depending on the dosage and timing. If you administer parathyroid hormone to bone tissue in a steady, continuous manner, this will lead to bone resorption and bone breakdown, and this will raise the serum calcium. And this is the way parathyroid hormone works physiologically to maintain calcium in our body. If, however, you artificially administer a low-dose, once-daily bolus of parathyroid hormone, you will see the opposite effect. The bone mass will go up and there will be bone formation. The reason that parathyroid hormone does this is not well understood, but because of this different effect of a once-daily bolus, you can use artificial parathyroid hormone. It's a drug called teriparatide to treat osteoporosis. You might think that giving parathyroid hormone to someone with osteoporosis, which is a disorder of weak bones, would be bad, but it turns out to be good because the once daily administration of a bolus works differently than the continuous steady infusion that the parathyroid gland does to the bones. So parathyroid hormone has different effects depending on whether you administer it continuously or as a single bolus. In addition, it affects the bone compartments differently. So cortical bone, which is the hard outer shell bone, decreases in response to continuous parathyroid hormone administration. So when your parathyroid gland releases continuous PTH, you see loss of cortical bone, and that's what raises the serum calcium level in response to parathyroid hormone from the parathyroid gland. Trabecular bone, on the other hand, increases in response to intermittent low-dose boluses of PTH. This is the trabecular portion of the bone here, and it's very useful that this effect of parathyroid hormone occurs on trabecular bone clinically because you can administer recombinant parathyroid hormone, that's teriparatide, and this will increase the bone density of trabecular spongy bone. And that's the type of bone that often decreases in osteoporosis. There's lots of it in the spine. There's also lots of it in the head of the femur. And these are places where people with osteoporosis have lots of fractures. So if you give recombinant parathyroid hormone, teriparatide, you will see a rise in the bone density of the trabecular bone, and it's very helpful for treating osteoporosis. Let's talk about how parathyroid hormone affects the different cell types in bone. Osteoblasts contain PTH receptors, and this is why low-dose intermittent boluses of PTH can increase bone mass. That's because the PTH can directly stimulate the osteoblasts. Osteoclasts have no PTH receptors. They are activated indirectly by osteoblasts, and this follows the principle we talked about before, that the osteoclasts are really slaves to the osteoblast. So the way that parathyroid hormone leads to breakdown of cortical bone and a rise in the serum calcium is by activating the osteoblasts, which in turn will activate the osteoclasts. And they do this through the mechanisms we talked about before. Osteoblasts can make MCSF and secrete that. They can also express RANK-L on their surface, and both of these will activate the osteoclasts, and that's how parathyroid hormone can lead to bone breakdown. I'll finish this video by activating the osteoblasts, which in turn will activate the osteoclasts, and they do this through the mechanisms we talked about before. Osteoblasts can make MCSF and secrete that. They can also express RANK-L on their surface, and both of these will activate the osteoclasts, and that's how parathyroid. To breakdown of cortical bone and a rise in the serum calcium is by activating the osteoblasts, which in turn will activate the osteoclasts. And they do this through the mechanisms we talked about before. Osteoblasts can make MCSF and secrete that. They can also express RANK-L on their surface, and both of these will activate the osteoclasts, and that's how parathyroid hormone can lead to bone breakdown. I'll finish this video by talking about estrogens. Estrogens have numerous effects on bone. First of all, they close the growth plate at puberty. So in girls at puberty, the ovaries begin making lots of estrogens, and that will close the growth plate. In boys at puberty, the testes make lots of androgens, but they also make estrogens, and those estrogens will also close the growth plate in boys, just like in girls. A second important effect on bone of estrogens is that they raise bone density, and this is why women often develop osteoporosis at menopause, because their ovaries stop making estrogen, estrogen levels fall, and that leads to a fall in bone density, and that leads to osteoporosis. The way that estrogens raise bone density is by decreasing the activity of osteoclasts. Remember, there's always a balance between bone formation and bone breakdown. If you decrease the amount of bone breakdown by osteoclasts, you will get relatively more bone production, and that's what leads to increased bone density. Estrogens have been shown to decrease osteoclast activity and even induce apoptosis of osteoclasts. And the way they do this is through those same signaling systems we talked about earlier. Estrogens have been shown to stimulate OPG synthesis by osteoblasts. Remember, OPG is the decoy receptor for RANK-L, so more OPG leads to less osteoclast activity. In addition, estrogens have been shown to decrease MCSF release by osteoblasts. They've also been shown to decrease RANK production by the osteoclast so that the receptor for RANK-L is not present. And through all these mechanisms, you get less activity of osteoclasts, and that's why estrogens are good for bone formation. And that concludes our video on bone.
How do you do that, bro? How the hell did you do that? How do you do that, man?
Hello, everyone, and welcome to our module on bone disorders. In the video on bone biology, I talked about how our bone is constantly turning over, meaning that osteoclasts are breaking down old bone and osteoblasts are laying down new bone. And if you understand that, then it's very easy to understand the bone disorders we'll talk about in this video. So osteoporosis, which I'm going to talk about in another video, is thinning of the bones. And this occurs when there is more bone breakdown relative to bone formation. Osteopetrosis is thick bones. In this condition, there's not enough bone breakdown and too much bone formation, and the bones become thick. In rickets and osteomalacia, these are both nutritional bone disorders where there's not enough calcium around and therefore bone cannot be laid down normally. And then finally, Paget's disease, which we'll talk about in this video, is a disorder where there's too much bone turnover. Let's start by talking about osteopetrosis. In this disorder, there is defective osteoclast activity, and this leads to a situation in terms of bone turnover where osteoblastic activity is more than osteoclastic activity. As a result, there is more bone laid down and there is an increase in bone density. There are a number of forms of this disorder, and you don't need to know all the clinical details for step one, but you should be aware that there is an autosomal recessive form or infantile form that is the most severe form of the disorder. This form presents in infancy, that's why it's called the infantile form, and it's often caused by a genetic defect in a gene that codes for the enzyme carbonic anhydrase. This enzyme is required to normally make acid, and recall that osteoclasts have to make acid in order to break down bones. When they can't do that, they can't break down bone normally. In addition, the kidney has trouble secreting acid, so these children often have a renal tubular acidosis. And then, as I said before, this is a very severe disorder, so these children also can have seizures and intellectual disability. There's also an autosomal dominant. Dominant form, which is called Albers-Schonberg disease. This can present in adolescence. It's a milder form of the disease, and sometimes it's asymptomatic, and it's identified because an X-ray is done for another reason, and evidence of osteopetrosis is found. You might think that in osteopetrosis, because the bones are thick, that they would be super strong and never break, but this turns out not to be the case. The bones in osteopetrosis are prone to fracture. There's a balance in our bones between strength and flexibility, and in osteopetrosis, they lose their flexibility and therefore they easily break. In addition, you can see excess bone laid down in the medullary cavity of the long bones, and this can lead to loss of the bone marrow. That can potentially cause pancytopenia. And when the bone marrow isn't making enough blood cells, the liver and the spleen can begin to make blood cells. This is called extramedullary hematopoiesis, and for this reason, in severe cases of osteopetrosis, you can see an enlarged liver and spleen. There can also be excess bone in the skull, and this can narrow the pathways by which cranial nerves exit the skull and lead to cranial nerve compression. So you can potentially see vision loss, deafness, or facial paralysis. And then finally, if there's bone deposition that narrows the foramen magnum, this can lead to hydrocephalus. It's very easy to recognize classic examples of osteopetrosis by X-ray. That's because the bones will be bright white. On the left side of the screen is a normal pelvis X-ray. On the right side of the screen is an X-ray from a patient with osteopetrosis. Notice how bright white all the bones are. Osteopetrosis is potentially curable with a bone marrow transplant. This is sometimes done for the more severe infantile form. Why is this the case? Well, that's because osteoclasts in our bones are derived from monocytes and macrophages. And here's a couple of monocytes on the screen to remind you of this. So if you replace the bone marrow and you replace the sick osteoclast with normal osteoclasts, the bone turnover problem will go away and bones may return to normal. Moving on, let's talk about rickets and osteomalacia. These are both nutritional bone disorders. They are caused by either low calcium or low vitamin D intake. They're usually seen in the third world in settings of famine and starvation, like this little boy shown on the screen here. In rickets and osteomalacia, there is poor mineralization of osteoid. So let's talk about what osteoid is. Osteoid is non-mineralized bone matrix. It's mostly proteins, and it's secreted by rickets bone disorders. They are caused by either low calcium or low vitamin D intake. They're usually seen in the third world in settings of famine and starvation, like this little boy shown on the screen here. In rickets and osteomalacia, there is poor mineralization of osteoid. So let's talk about what osteoid is. Osteoid is non-mineralized bone matrix. It's mostly proteins, and it's secreted by osteoblasts when they want to make new bone. Later, it becomes mineralized with calcium and phosphate. So in rickets and osteomalacia, the osteoid cannot be mineralized, but it keeps being laid down by osteoblasts. So where are you going to see osteoid? It's going to be at sites of new bone growth. In children, that will be at the growth plates, and in children and adults, that will be at sites of bone turnover. So in rickets, you have problems with osteoid deposition at the growth plates. In children and adults, you can see osteomalacia, which is problems with mineralization of osteoid at sites of bone turnover. There is poor mineralization of osteoid. So let's talk about what osteoid is. Osteoid is non-mineralized bone matrix. It's mostly proteins, and it's secreted by osteoblasts when they want to make new bone. Later, it becomes mineralized with calcium and phosphate. So in rickets and osteomalacia, the osteoid cannot be mineralized, but it keeps being laid down by osteoblasts. So where are you going to see osteoid? It's going to be at sites of new bone growth. In children, that will be at the growth plates, and in children and adults, that will be at sites of bone turnover. So in rickets, you have problems with osteoid deposition at the growth plates. In children and adults, you can see osteomalacia, which is problems with mineralization of osteoid at sites of bone turnover. To understand rickets, let's review what growth plates are. They're found at the ends of long bones in children. If you look at this x-ray on the screen here, this is the growth plate from a long bone in the arm of a child. The growth plates contain hyaline cartilage, and there are chondrocytes in the growth plate that grow toward the epiphysis. So that's this direction here. In the other direction, going this way, osteoblasts are laying down matrix. That's towards the diaphysis, or the straight part of the bone. And this is how the long bones get longer in children, and that's why their arms get longer and their legs get longer and they get taller. In rickets, the growth plates in children will thicken. This is because the chondrocytes continue to expand, although they display disorganized growth. And the osteoblasts lay down osteoid only. The osteoid does not become mineralized, so it just builds up, and the bone thickens from osteoid accumulation. And for this reason, you can see weakened bones and distorted bone growth, and you can also see thickening of the bone around the growth plate. Some of the clinical findings of rickets are shown in this slide. You can see epiphyseal widening of the long bones. The epiphysis is the end of the long bone right here, and it can become widened from osteoid deposition. You can also see bowing of the legs. This is called genu band, although they just get longer in children, and that's why their arms get longer and their legs get longer and they get taller. In rickets, the growth plates in children will thicken. This is because the chondrocytes continue to expand, although they display disorganized growth, and the osteoblasts lay down osteoid only. The osteoid does not become mineralized, so it just builds up, and the bone thickens from osteoid accumulation. And for this reason, you can see weakened bones and distorted bone growth, and you can also see thickening of the bone around the growth plate. Some of the clinical findings of rickets are shown in this slide. You can see epiphyseal widening of the long bones. The epiphysis is the end of the long bone right here, and it can become widened from osteoid deposition. You can also see bowing of the legs. This is called genu varum. I'll show you a picture of this in a minute. You can also see swelling at the costochondral junctions in the sternum. This can create a finding called the rachitic rosary, which I'll show you a picture of in a minute. And then in the skull, you can get craniotabes, which means a soft skull. The occipital and parietal bones are particularly vulnerable to this. This is the occipital bone, which I'll show you a picture of in a minute. I'll show you a picture of this in a minute. You can also see swelling at the costochondral junctions in the sternum. This can create a finding called the rachitic rosary, which I'll show you a picture of in a minute. And then in the skull, you can get craniotabes, which means a soft skull. The occipital and parietal bones are particularly vulnerable to this. This is the occipital bone here. This is the parietal bone in blue. They can become soft and collapse with pressure in children who have severe cases of rickets. Here's a picture of epiphyseal widening. You can see this child's wrist is swollen. That's because the epiphysis is accumulating osteoid. The growth plates in children with rickets can become abnormal. On the left side of the screen is a normal growth plate. On the right side of the screen is one from a patient with rickets. You can see that the growth plates are much wider than normal. You can also see that the bones are frayed and irregular. This is all evidence of rickets. And here's an example of the bowed legs of children with rickets called genu varum. Basically, the wider the growth plates in children with rickets can become abnormal. On the left side of the screen is a normal growth plate. On the right side of the screen is one from a patient with rickets. You can see that the growth plates are much wider. The normal, you can also see that the bones are frayed and irregular. This is all evidence of rickets. And here's an example of the bowed legs of children with rickets called genu varum. Basically, the bones are weaker than normal, and the child's weight presses down on them, so they become bowed like this, a classic finding in cases of rickets. Your ribs meet cartilage. In the front of your chest at a location called the costochondral joints or costochondral junction. And the ribs can become swollen at this point in children who have rickets. And this can lead to little bumps, as seen in this child on the left side of the screen. You can also see the bumps here on this x-ray. And this is called a rachitic rosary. A rosary is a necklace with beads, and these look like beads on the rosary. So rickets is caused by a nutritional deficiency, but you will only see rickets in children because only children have growth plates. Osteomalacia, which is also a bone disorder caused by nutritional deficiency, can occur in children or adults. It doesn't occur at the growth plates. Instead, it occurs in areas of bone turnover throughout the body. It often presents as bone pain or tenderness. So if you were to go to a third world country and be subjected to famine and your bones began to ache, this would be the reason why. It's often in the spine, pelvis, and legs, and the bones become generally weak and they are prone to fractures. There are two classic X-ray findings of osteomalacia. One's called a pseudofracture. The other is called a loser zone throughout the body. It often presents as bone pain or tenderness. So if you were to go to a third world country and be subjected to famine and your bones began to ache, this would be the reason why. It's often in the spine, pelvis, and legs, and the bones become generally weak and they are prone to fractures. There are two classic X-ray findings of osteomalacia. One's called a pseudofracture. The other is called a loser zone, named for the person who first described it. These are caused by repaired stress fractures that are inadequately mineralized. They're also caused by erosion of bone by arterial pulsations. So the bone becomes weak, and when your arteries pulse with blood flow, that can erode the bone around the arteries. This is a pseudofracture shown here. You can just see this white line in a long bone. And this is a loser zone here. It just looks like a black space in a long bone. So I told you that rickets and osteomalacia are due to vitamin D and calcium dietary deficiency. So let's talk about that in a little more detail now. Usually these disorders are caused by a vitamin D deficiency, which in turn leads to a calcium deficiency. So what are some of the reasons a person could be vitamin D deficient? Well, if a mother doesn't take in enough vitamin D during pregnancy, her baby can be born with signs and symptoms of rickets and osteomalacia. Also remember that one of the major sources of vitamin D besides your diet is from the skin, from sun exposure. So if a person doesn't get enough sun exposure and they don't eat enough vitamin D in their diet, they can become vitamin D deficient. Vitamin D is also a fat-soluble vitamin. So if a person has fat malabsorption, they can become vitamin D deficient. So you can see fat malabsorption in cystic fibrosis, pancreatitis, celiac disease, inflammatory bowel disease. All of these things could potentially lead to vitamin D deficiency and rickets and osteomalacia. And then finally, you need your liver and your kidneys to make the active form of vitamin D. So if a person has liver or renal failure, they cannot make enough active vitamin D, and that can lead to rickets or osteomalacia. You can one of the major sources of vitamin D besides your diet is from the skin, from sun exposure. So if a person doesn't get enough sun exposure and they don't eat enough vitamin D in their diet, they can become vitamin D deficient. Vitamin D is also a fat-soluble vitamin. So if a person has fat malabsorption, they can become vitamin D deficient. So you can see fat malabsorption in cystic fibrosis, pancreatitis, celiac disease, inflammatory bowel disease. All of these things could potentially lead to vitamin D deficiency and rickets and osteomalacia. And then finally, you need your liver and your kidneys to make the active form of vitamin D. So if a person has liver or renal failure, they cannot make enough active vitamin D, and that can lead to rickets or osteomalacia. You can also develop these disorders from calcium deficiency in absence of a vitamin D deficiency, but this is very rare. Usually people are deficient in vitamin D and secondarily deficient in calcium. You only see this if a person takes in very, very low amounts of calcium, but also happens to get enough vitamin D from the sun or their diet. So you can imagine, knowing what's on this slide, that the treatment of these disorders is very straightforward. They are treated with vitamin D supplementation and calcium supplementation. On your step one exam, they like to ask you about the lab findings in rickets and osteomalacia. So people with these disorders have low serum calcium levels. Obviously, that's what leads to the problem mineralizing osteoid. And usually they also have a low vitamin D level because, as I told you before, these disorders are almost always due to a vitamin D deficiency. Let's talk about vitamin D in a little more detail. So there are two sources for vitamin D, your diet and your skin. Both of them can make an inert form of vitamin D, which then has to be activated by the liver and the kidneys. The liver hydroxylates vitamin D to make 25-hydroxyl vitamin D or calcidiol. The kidneys hydroxylate the vitamin again to make 125-hydroxyl vitamin D, which is called calcitriol, and this is the active form of the vitamin. Now, that 25-hydroxyl form of vitamin D made by the liver is the storage form. It's constantly produced by the liver, and it's available in the body for activation by the kidneys as needed. This means that the best test to assess vitamin D status is the serum 25-hydroxyl vitamin D level. This form of vitamin D has a long half-life, and the liver production of this form of vitamin D is not regulated by PTH. So if it's low, it means you are not getting enough vitamin D from your diet or your skin. If it is normal, it means you are getting enough from your diet or your skin. The active form can go up and down depending on your parathyroid hormone level, but this is not true for the 25-hydroxyl storage form. Other findings in rickets and osteomalacia are shown here. The parathyroid hormone level will be high. This is the normal response of the parathyroid gland to low calcium levels, which you see in rickets and osteomalacia. There will be a low serum phosphate level. This is because the high PTH promotes excretion of phosphate by the kidneys. And then finally, there will be a high level of bone alkaline phosphatase. This enzyme level reflects osteoblast activity, and the osteoblasts are working very hard to lay down osteoid. They just can't lay down mineralized bone. Our next disorder is Paget. Disease, which is also called osteitis deformans. This is a focal disorder of bone. So this is distinct from osteoporosis, osteopetrosis, and rickets and osteomalacia. Those disorders affect all the bones. Paget's disease develops focally, often in just a single bone. And this is a common disorder in older patients. The average age of diagnosis is about 70 years old. Paget's disease is a disorder where there is excessive bone remodeling or bone turnover. When this happens, there's overgrowth of bone at focal sites within individual bones, and the new bone that is laid down is abnormally large and deformed. The exact cause is unknown, but the primary cause is believed to be abnormal osteoclasts. Remember, osteoclasts are the cells that break down bone, and in Paget's disease, it's believed that they begin breaking down bone, and then osteoblasts lay down new bone, and you get excessive remodeling, and that leads to these focal sites of overgrowth of bone. Paget's disease evolves through a number of phases or stages. The first stage is called the osteolytic stage. This is the stage where osteoclasts are breaking down bone. And under the microscope, you can see bizarre shaped osteoclasts. There are many of them. They are multinucleated, and these are involved in the osteolytic phase of Paget's disease. The second phase is called the mixed phase. That's because there is osteolytic and osteoblastic activity. In this phase, osteoblasts have begun laying down new bone. And then the final phase is called the osteosclerotic phase. This is where bone formation dominates, and you get this thickened abnormal bone present at focal spots within individual bones. Also in this phase, there is a hypervascularity that occurs because the bone has lots of blood vessels in it. And this will be important in a minute when we talk about some of the potential complications of Paget's disease. Under the microscope, the hallmark of Paget's disease is a mosaic pattern of lamellar bone with cement lines. So on the right side of the screen is normal lamellar bone. It forms these nice even lines. On the left side of the screen is bone from Paget's disease. You can see that it's all distorted. That's the mosaic pattern. There are also these lines called cement lines that are laid down. These are the classic findings. Under the microscope, the hallmark of Paget's disease is a mosaic pattern of lamellar bone with cement lines. So on the right side of the screen is normal lamellar bone. It forms these nice even lines. On the left side of the screen is bone from Paget's disease. You can see that it's all distorted. That's the mosaic pattern. There are also these lines called cement lines that are laid down. These are the classic findings in Paget's disease. Paget's disease may be asymptomatic. It's sometimes picked up when an X-ray is done for another reason and evidence of Paget's disease is seen. It often affects the long bones, but also the skull and the spine. And the most common complication is a pathologic fracture, classically a chalk stick fracture, where the bone breaks like a piece of chalk. I'll show you a picture of this in a minute. Patients can also have bone pain from microfractures within the bone, and sometimes patients who have Paget's disease of the long bones of the legs can develop bowing of the legs. This is a picture of a chalk stick fracture of the femur. You can see that the bone has just snapped like a piece of chalk. This is a pathologic type of fracture, often seen when there's something wrong with the underlying bone, like Paget's disease. Other findings of Paget's disease are shown on the slide here. Patients can develop an enlarged skull. Men who wear hats, which is very common these days, but if they do, can complain of an increased hat size. Cranial nerves can become compressed from skull enlargement. This can lead to deafness potentially. You can see radiculopathy at the spine if the bones of the spine compress a nerve root. And sometimes you can see erythema over the affected bone, especially when it's in the leg or one of the bones of the arms. You can see redness over the swollen bone. And this is due to the hypervascularity of the bone. So there's a rare complication of Paget's disease that they love to ask you about in step 1 of your boards, and that is high output heart failure. All that increased vascularity of the bone can lead to a connection between the arterial side of the vascular system and the venous side of the vascular system. This is called an AV fistula in the new bone. And when you have an AV fistula, blood bypasses the resistance vessels like the arterioles, and this can lead to high output heart failure. A rare but high-yield complication of Paget's disease to know about. Another complication you should be aware of is osteosarcoma. This is a rare bone tumor that I talk about in another video, but patients who have Paget's disease are at increased risk for this bone tumor. The classic lab finding of Paget's disease is an increased bone alkaline phosphatase level. This reflects the increased activity of the osteoblasts. And you can treat Paget's disease with bisphosphonates and calcitonin, which are 2 drugs also used to treat osteoporosis. And I talk about their mechanism of action and side effects in the video on osteoporosis drugs. Osteitis fibrosa cystica is the classic bone disease seen in hyperparathyroidism. I talk about hyperparathyroidism in the endocrine section in the video on the parathyroid gland, but I'll review the bone features of hyperparathyroidism here. So the clinical features of osteitis fibrosa cystica are bone pain and fractures. The high levels of PTH lead to bone resorption, and that leads to thinning of bones and weak bones. The classic cause of this condition is a parathyroid adenoma. This will lead to high PTH levels that will promote bone breakdown and lead to osteitis fibrosa cystica. It will also lead to hypercalcemia. The phosphate level will be low. However, that's because parathyroid hormone promotes phosphate excretion by the kidneys. The 2 classic X-ray findings of osteitis fibrosa cystica are subperiosteal bone resorption and Brown tumors. Parathyroid hormone promotes thinning of the cortical or hard bone, and this sits right below the periosteum as shown in this picture here. So subperiosteal bone resorption is commonly seen in the bones of the fingers. What you will see are irregular or indented edges to the bones. Brown tumors are also called osteoclastomas. These are collections of giant osteoclasts in the bone. Parathyroid hormone promotes thinning of the cortical or hard bone, and this sits right below the periosteum as shown in this picture here. So subperiosteal bone resorption is commonly seen in the bones of the fingers. What you will see are irregular or indented edges to the bones. Brown tumors are also called osteoclastomas. These are collections of giant osteoclasts in the bone. They're mixed with stromal cells and matrix proteins, and they appear as big black spaces in the bone on X-ray. And shown on this slide are a couple of examples of brown tumors. You can see the red arrows on the left are pointing to black spaces in the long bones. Those are brown tumors. You can also see the white arrows on the right pointing to these big black spaces in the bones of the fingers. Renal osteodystrophy are pointing to black spaces in the long bones. Those are brown tumors. You can also see the white arrows on the right pointing to these big black spaces in the bones of the fingers. Renal osteodystrophy is a term that refers to the bone abnormalities that are seen in patients with renal failure. They can develop many of the bone problems we've talked about in this video. So patients who have renal failure are hypocalcemic. This is because the kidneys cannot make the active form of vitamin D, and therefore they become hypocalcemic. When the calcium is low, they develop hyperparathyroidism. So these patients can have problems from low calcium and from high PTH levels. So they can develop osteitis fibrosa cystica because their PTH level is high. They can develop rickets and osteomalacia because they are vitamin D deficient. They can develop osteopenia and osteoporosis, and this can lead to growth retardation in children. There can be bone pain and there can be fractures. This is all the constellation of bone problems that occurs in the setting of renal failure. And if you're ever given a tricky board question where they don't tell you the patient has renal failure, but they ask you for the cause of the bone problem, remember that when the PTH level is high, the phosphate level should always be low because PTH promotes excretion of phosphate. The only time you'll ever see the PTH level high and the phosphate level high is when the patient has renal failure and they can't get rid of phosphate. That will lead to high phosphate levels despite a high PTH level. I'll finish with this slide showing an algorithm for how to approach problems where patients have bone pain or easy fractures and you're asked to determine the cause. So first of all, look at the calcium and PTH levels. If they're both normal, then you're dealing with some primary bone problem. This could be osteoporosis, it could be osteopetrosis, it could be Paget's disease, or it could be a bone tumor. In all of these disorders, usually the calcium and PTH levels are normal. If they tell you that the calcium level is low and the PTH level is high, this is the normal response of the parathyroid gland to a low calcium level. So here the problem is with calcium, not the parathyroid gland. And this is usually a vitamin D deficiency. It may be a dietary deficiency, it could be not enough sun exposure, or it could be a malabsorption syndrome, but there's some problem getting enough active form of vitamin D in the body. If they tell you that the calcium level is high and the PTH level is high, this is abnormal in terms of the parathyroid gland. The PTH level should never be high when calcium is high. So this is telling you that the patient has a parathyroid problem. They have osteitis fibrosis cystica, and they likely have a parathyroid adenoma. And that concludes our video on bone disorders. Hello, everyone, and welcome to our module on osteoporosis. Osteoporosis gets its name because in this disorder,